Well, another amazing poster session upstairs, and just really very vibrant discussions, excellent multidisciplinary science. Professor Lumsden commented to me about how fantastic it is to have an interdisciplinary environment here, and I think this is one of the blessings we truly have in the Department of Surgery. So we're ending on a big highlight here. I think the trainee presentations really are truly at the essence of what we do here on this day, and the trainees are so important to the vibrancy of our department. So we will have Gallie, Bateman, McMurrick, and translational research presentations by trainees, and this session will be chaired by Dr. Chaya Schwartz, who is a surgical oncologist, HPB a surgeon, associate director of the Abdominal Transplant HPB Fellowship at UHN, and faculty in the Department of Surgery at University of Toronto. So Chaya, over to you.
Thank you very much. So we'll start with the first speaker, Dr. Koga, who is a postgraduate fellow for Dr. Yasufuko in his lab in the thoracic laboratories. He is a thoracic surgeon, and research interests is transnational research about generation of patient-derived lung cancer models. His talk will be about establishment of lung cancer organoids using endobronchial ultrasound-guided transbronchial needle aspiration specimen via xenograph tumor. Please.
Thank you for the introduction, and thank you for this great opportunity to have a presentation at this Gallie Day. Patient-derived organoid, we call it PDO, using cancer cell is a novel 3D culture preclinical model in cancer research. PDO recapitulates the characteristics of parental cancer, and PDO can predict the drug sensitivity of patients. These findings in several types of cancers are reported in many high-impact journals in the past 10 years. And PDO is a promising model for utilization in drug discovery and testing of treatment. Patient-derived lung cancer organoid, LCO, is also reported to recapitulate the parental cancer cell characteristics and demonstrated the ability to predict drug response. Long-term success rate in the 10 or more passages of establishment of lung cancer organoid is up to 80% with malignant pleural effusion, but only 7 to 70% with surgically rejected lung cancer specimen in previous reports, because normal epithelial cell outgrowths frequently occur in the rejected lung cancer specimen. And previous study about biopsy sample is very limited. We focused on the XDO, xenograft-derived organoid, because success rate of long-term culture of XDO was higher than LCO in surgically rejected samples in our previous study. In addition, PDX was successfully established from EBUS-TBNA samples in 42% with rapid on-site evaluation. As such, we were interested in the feasibility of EBUS-based XDO establishment. A total of 16 cryopreserved EBUS PDX tumors were engrafted in NSG mice. Rejected PDX tumors were digested to single cells, and tumor cells were embedded in matrigel and cultured in M27 medium developed at our laboratory. We defined the stable establishment of XDO as 10 or more passages, no mouse cell contamination by flow cytometry, and retained pathological feature of the original PDX tumor. 8 adenocarcinoma, 2 squamous, 3 small cell lung cancer and each one of LC neck and large cell carcinoma were included in this study. Many of them were advanced stage and 2 of them were stage 2 disease. These implanted PDX tumors successfully revived and reached 1,000 cubic millimeter in median 8.3 weeks. Total of 13 EBUS XDOs were passaged more than 10 times over a median time period of 14.4 weeks after processing PDX tumor. As shown, all EBUS XDOs expanded in small cluster formation, and it was very difficult to distinguish the pure cancer XDOs from the mouse cell outgrowth one based on the morphology. So flow cytometry analysis revealed that 12 XDOs were almost 100% enriched by human cells, except for an adenocarcinoma, which was completely mouse cell outgrowth. EBUS XDO and original PDX showed similar histological appearance and maintained the expression of TTF1 in adenocarcinoma and P40 in squamous cell carcinoma. This marker expression was consistent with the paired EBUS samples based on the findings in the pathological report. They are unique to adenocarcinoma XDOs. The 4370 model is a lung mucinous adenocarcinoma characterized by mucinous component and maldistributed nuclei. We can observe these findings in both XDO and PDX. And 534 is an adenocarcinoma with neuroendocrine features. Both XDO and PDX strongly expressed neuroendocrine markers. Small cell EBUS XDO and LC-neck XDO models both showed strongly expressed expression of the neuroendocrine markers. In summary, we successfully established total of 12 stable EBUS XDOs in 80% success rate according to our criteria. Driver mutation status was also concordant across the XDO and PDX. Sanger sequence showed that heterozygous sequence of the KRAS G12C mutation in 586 PDX model and homozygous sequence status of 555 model were recapitulated in the XDO models very well. To check the utility of EBUS XDOs for drug sensitivity assay, we compare the drug response of the EBUS XDO and its patient. The patient of 517 who had a stage 4 small cell lung cancer disease responded to the carboplatin and etoposide treatment very well. Consistent with it, XDO model showed sensitivity to the etoposide and carboplatin treatment, and its IC50 values were lower than the XDO 4370 adenocarcinoma model, whose patient showed a stable disease to the cisplatin and etoposide treatment. XDO 586 harbors the KRAS G12C mutation that is one of the most popular driver mutation in lung cancer, and I checked the sensitivity to KRAS G12C specific inhibitors, sotorasib and adagrasib. This model showed a good response to this agent in drug sensitivity assay compared to the XDO344 resistant model. And in killing assay, the cell growth of 586 model was suppressed by the treatment of sotorasib and adagrasib. In immunoblotting, showed that downstream signals of XDO856 was suppressed by dose-dependent manner of the KRAS G12C inhibitor treatment. Using this sensitivity model, we generated resistant clones by chronic exposure to these KRAS G12C inhibitors. We successfully established resistant clones, and they showed much higher IC50 values compared with parental models, and cell number increased under treatment by KRAS G12C inhibitors. We are currently looking into the resistant mechanism by multiomics analysis. In conclusion, we successfully established EBUS PDX derived organoid in 80% success rate through careful validation. Our data suggests that XDO can be a good option to generate a stable organoid from biopsy specimen of the advanced lung cancer. XDO is also available as in vitro model for the insight to acquire the resistant mechanism to target therapy. We are providing long-term EBUS XDO models established in this study to Princess Margaret Living Biobank. We are just analyzing the genomic, transcriptomic, and proteome data, and the data should be integrated to digital data set. I'm very happy if UHN researchers will be interested in our model and will use them in their research. I appreciate members of YASF Club and Tau Lab for all of their help, and thank you again for this great opportunity of Gallie representation. Thank you very much.
Thank you very much for an excellent presentation. Dr. Jennifer Chang.
Hi, I'm Dr. Chang. I'm a cardiac surgeon here at TGH. Thank you for the opportunity to discuss this paper, Dr. Koga. First, congratulations. I'm not a cancer surgeon or a cancer researcher, so it was actually very exciting for me to read this paper and learn about all the fantastic work that's being done in other parts of this department.
Thank you.
So first, when I was reading the paper, thank you for sharing it. Previous studies, only 10% or 7 to 17% of the surgical samples were able to be successfully established LCOs, and you guys were able to achieve 75% success rate. So I was wondering what you think the magic is. Is it the EBUS that allows you to decrease contamination of other types of cells, or is it the xenograft culture techniques that your lab has developed?
Thank you for the question about that point. Yeah. Actually, when we culture the organoids with surgically rejected sample, normal airway cell outgrowth frequently occurs, and that affects the low establishment rate when we use a rejected sample. And EBUS PDX sample is extracted the normal cell contamination during the PDX and organoid culture. So that's why we get a high success rate using the EBUS PDX sample, EBUS XDO sample.
Okay, thank you. My second question is, you also broke down your samples in terms of the different types of cancers, and the adenocarcinoma group had a slightly lower success rate. Are you expecting different success rates depending on the different cancer types, or this is a statistical thing just because there aren't so many in the other types?
Thank you. In our experience and previous data, the success rate of adenocarcinoma is success rate of organoid culture with adenocarcinoma is usually a little higher than squamous cell, but lower than the small cell lung cancer. So our data our data is usually consistent with the previous data, I think. Yeah.
Okay. And then my final question is if you could, is more speculative, and if you could paint a picture of how you would like to see the organoids used in clinic, in terms of personalized medicine, or how would you envision this being used?
If possible, I'm expected the patient didn't want to do the personalized medicine, but the problem is the time course of the establishment of these models. So that's a matter that we should discuss in future research, I think.
Okay, thank you very much. Okay.
Thank you very much. An excellent presentation.
Thanks so much.
Our next presenter will be Dr. Malak El-Batarni. She is a third-year cardiac surgery resident at the University of Toronto and a second year in their PhD program. She's also part of the SSTP program and doing a lot of global surgery. The title of her presentation will be Integrative Phosphoproteomic Analysis Reveals Human Thoracic Aortic Aneurysm Differ by Anatomic Segment, Conventional and Machine Learning Analysis. Please.
Thank you very much, Dr. Schwartz, and thank you for the opportunity to present this work. We'll be discussing thoracic aortic aneurysm and methods of biochemical and bioinformatic profiling. And so the disease we're interested in is really type A dissection and thoracic aneurysm. And while we can deal with thoracic aneurysm in the elective setting without too much difficulty, type A dissection remains a high mortality condition with about 15 to 30% mortality even in the modern era. And this affects also a high proportion of young patients. Unfortunately, our strategies for picking which patients to operate on remain poor predictors, namely diameter of the aorta. And in fact, close to half of all dissections are occurring at subsurgical threshold diameters. And so it goes without saying that not all aneurysms are really created equally, and prognostication remains a significant challenge. While half of dissections are occurring in small aneurysms, many small aneurysms actually do not progress. And one of the things, there are many factors that impact progression of thoracic aneurysm, and one of those factors is actually the anatomic segment. And so here I'm referring to whether the root, ascending, arch, or descending thoracic aorta are affected by aneurysm. And to the moment, we're still unclear as to what biologic mechanisms are actually underpinning these differences, whether they're intrinsic tissue properties, hemodynamics, or other factors. And so that's where we bring in mass spectrometry proteomics. And this is a highly sensitive, very precise tool for phenotyping tissues. And importantly, it's closer to the phenotype at the protein level than DNA or RNA profiling. And so when we combine mass spectrometry proteomics with high throughput of samples, it actually facilitates resolution of phenotypic differences even within different segments of the same organ. Additionally, proteomics allows us to examine post-translational modifications, things like protein phosphorylation, which can drastically alter protein function without changing the overall level. And so this again brings us one step closer to understanding disease phenotypes. So our objective was quite simple. We wanted to compare the phenotypic profile of human aortic tissue samples among these different anatomic segments with a particular focus on root versus ascending and also descending regions of aorta. And we're using a label-free mass spectrometry proteomic approach, as well as an additional phosphoproteomic approach requiring multi-step phosphopeptide double enrichment. And these are both methods which we customized and optimized in-house. And I'll spare this audience the details of that, but happy to talk in detail if you wish. And basically, the distinction here is that this is comprehensive profiling of the entire set of proteins captured in a particular sample and every single phosphorylation site on every single protein in a sample, as opposed to a panel of genes or a panel of RNA transcripts. And so we included 148 human tissue samples from 82 unique individuals, and the breakdown of different regions is included here. The majority of our samples came from elective aneurysm patients, but we also had type A dissection and also a subset of controls. And the overall clinical characteristics are represented here, and really the main message is that age, gender, and medical comorbidities were not significantly different between patients. There were some differences, of course, in surgical procedure owing to the different region of aorta that it's being removed. However, importantly, we had over 30 within patient controls where multiple segments are represented from the same patient. So our analytic approach involved initially comparing aortic samples by aortic segment directly. We also considered a subgroup of the aneurysm-only group. We then did a dimensionality reduction analysis to identify clusters of samples that were most similar to one another. And we also analyzed the biologic relevance of significantly different proteins to understand what the actual function was of these differences. And then we did some validation using immunofluorescence and immunoblotting, which I will touch on a little bit today. So starting first with the global proteomic profile, across all of the samples that we analyzed, we were able to identify over 5,600 proteins. And in comparison, this is several hundred to 1,000 proteins more than previously published vascular literature. And we identified a large number of significantly different proteins between the different segments, in particular between the root and descending, root and ascending, as well as the clicker is, there we go, as well as between the root and ascending segments. And so we focused our in-depth analyses more on these comparisons. This is an example of the aneurysm-only subgroup. So each of these dots in red represents a significantly different protein. And as you move up in the y-axis, it becomes a more significant difference. And you can see, again, there's a significant spread of significantly different proteins between these different segments when considering the aneurysm-only subgroup. We then wanted to understand whether samples were clustering independently in accordance to their proteomic profile, and whether this was in accordance also with the aortic segment. And so we found indeed with unsupervised clustering using principal component analysis. Hopefully you can appreciate each one of these squares represents a single sample, and they're clustering in accordance with their proteomics and in accordance with their segment of origin, with clear separation between root and descending, ascending and descending, and also modest clustering between the ascending and root. We also found similar results when performing this analysis using TISNE, which is another strategy for dimensionality reduction. And then we wanted to look at the phosphoproteomic profile of these tissues. So I don't have a comparator because this is sort of the first phosphoproteomic study in human aneurysm, but we were able to cover 1,400 phosphorylation sites in all of our samples. This is just a taste, but looking at root versus ascending, we found a number of significantly different phosphorylation sites. And importantly, these were only significant at the phospho level, and the overall level of protein was not different, suggesting that the difference was occurring as a result of phosphorylation. So we were quite excited about this. We wanted to understand what the relevance of significantly different proteins and phosphorylation sites was between different segments. And so here, focusing on root and ascending, we see both analyses reveal differences in cellular adhesion, binding, and developmental processes. At the protein level, metabolic processes were also highlighted. And at the phospho level, scaffold proteins and cytoskeletal regulation were significantly altered. We performed an integrative phosphoproteomic and proteomic gene set enrichment analysis to understand directionality of changes. And you can see that cytoskeletal organization, cellular adhesion, developmental processes were increased in ascending tissues, while mitochondrial energy and metabolism in root tissues. So to begin with validation, this is just a taste, but one of the proteins we identified was significantly different between segments was MFAP4. And interestingly, this protein has been previously published as a potential biomarker where serum MFAP levels actually were associated with type B dissection risk, which affects the descending, but not proximal risk of intervention. And really, there was no specific explanation for this initially. But in our data, as mentioned, we found significantly higher expression of MFAP4 in the ascending tissues compared to root and descending. And we validated this using immunofluorescence, so hopefully you can appreciate that there's increased expression significantly different and higher in the ascending group. We also localized MFAT4 expression to the vascular smooth muscle because we observed these differences specifically in the tunica media. And you can see that MFAT4 co-localizes with alpha SMA, smooth muscle marker, and not fibroblast or endothelial markers. So in summary, we've so far established the largest thoracic aortic tissue bank for multiproteomic analysis to date. We've developed a customized protocol for mass spectrometry proteomics with enhanced depth of coverage in compared to vascular literature by hundreds to thousands. We've performed the first phosphoproteomic analysis of human thoracic aneurysm, and we did so at a large scale. And we performed the first systems biology comparison of thoracic aneurysm on the basis of anatomic segment. We also confirmed segmental differences in a proposed biomarker, MFAT4, and we offered this as an explanation for why it has had inconsistent predictive value. Certainly many limitations, including lower representation of arch and descending segments in our sample population, dealing with batch effects, and also we're not quite powered to compare subgroups on the basis of valve morphology or genetic diagnoses just yet, but we're getting there. And for us as clinicians, the key messages really are that thoracic aneurysm certainly requires major surgery and is, prognostication remains a major problem. We've confirmed that thoracic aneurysms are biochemically distinct at different segments and therefore should be treated as such. Phosphoproteomics and proteomics may explain these differences in natural history and outcome, which we see all the time in the clinic. And any potential biomarkers that would be raised for thoracic aneurysm may be confounded by aortic segment. So this is something important to keep in mind. And lastly, I think we can all agree that there's a strong need for patient-tailored aneurysm management, and this should be on the basis of careful biochemical profiling. I have many people to thank, not least of which my co-PIs, Dr. Zunian and Dr. Gramellini, and all of the members of my lab, as well as our funding agencies. Thank you very much.
This is really amazing work. Dr. Forbes.
Yeah. So thanks very much. That was excellent work. Congratulations to you and your collaborators. So I like this presentation because it really brings biology into something where we often take a pretty simplistic approach, right? We know that diameter is the simplest thing to measure, but it's probably not as predictive of adverse outcomes as we think it would be. So I have a couple questions for you. And I love the term proteomic signature of the aorta, right from the valve all the way down. So how do your findings relate to the embryology of the aorta?
Yeah, great question. And it's sort of one of the inspirations for this project in the first place is the fact that different segments of aorta do come from different developmental origins. And interestingly, a lot of our pathway analysis did reveal some of these developmental pathways being altered in different segments of aorta. So, one of the things that has been proposed is that an initiating factor for dissection is actually the interface between different developmentally distinct regions of aorta. And so I think understanding the developmental pathways that are leading an aneurysm to progress is not only going to help us to identify specifically which regions of aorta are going to be most at risk, but also hopefully, of course, developing therapeutics in the future.
Great. And my other questions are around the fact that your pathology specimens are restricted to patients who have either had symptoms, they've had a type A dissection, or they've met a certain threshold of diameter, right? They're not just by nature of the investigation, they're not pre-repair specimens. So how much of the differential findings that you found between say, the ascending aorta and the descending might be reflected by the different pathologies? In type A, in the ascending aorta, probably predominantly symptomatic acute type A dissections, whereas in the descending, it's asymptomatic large aortas. Would that be somewhat of an issue?
Yeah, I think you raise some very important points, and it's so tricky to get access to that group of aneurysms that are at risk but haven't yet met surgical thresholds. And so one of the things that we're potentially hoping to do is there is currently a clinical trial underway, a Titan SVS study, which is randomizing patients to watchful waiting at subsurgical thresholds or surgery. And so really it would be fantastic to be able to get access to, to compare those samples to see if what we're seeing is occurring at the early at-risk state or not. And the other thing is we've been collecting blood samples on all these patients because ultimately whatever we find at the tissue level is not going to be clinically useful because we can't get it until the patient has already had surgery. So what we're working on right now is some of these proteomic and phosphoproteomic targets, developing ELISAs for them and being able to see whether they actually can be detected in blood and whether they can be predictive in a progressive fashion of which aneurysms are most likely to dissect.
So, and that's a nice segue into my last question. So you have a patient in front of you. Currently, right now, it's very easy to measure a diameter and give sort of a treatment recommendation. But in the future, I mean, your group has done a lot of work, diameter, CFD, biology here. How do you think you're going to incorporate all that so you can talk to the individual patient about a personalized therapeutic plan?
Yeah. So what I've presented today is actually just a subset of my overall PhD thesis aims, and really what we're doing is multi-omic analysis. And so we have accumulated a large cohort of samples for actually whole genome sequencing as well as transcriptomics. We have imaging data on many of these patients, and now we're working with the group with Dr. Bo Wang and the Vector Institute to perform basically multi-omic integration using AI analyses and figure out what the predictive factors are to be able to elucidate different phenotypes of aneurysm, and hopefully one day be able to go into the clinic and say, input all of these clinical variables, imaging, blood tests, this sort of thing, and have at least a better idea of maybe which individuals require immediate therapy or which could be waited for more safely.
Our next speaker is Hilary Leah. She's a MD PhD student at the University of Toronto under the supervision of Dr. Caroline Moulton and Dr. Frank Rudzik. She has a very high interest in medical education, and come on. Her title of the presentation: the role of operating room professions in setting the tone for teamwork, a constructivist grounded theory study.
Thank you for the introduction, Dr. Schwartz, and thank you to the organizing committee for the opportunity to present today. So as you know now, my name is Hilary Leah. I'm an MD PhD student here at the University of Toronto, and I'll be providing a brief summary of our work examining team dynamics by formalizing the construct of tone in the operating room. And so to start off with, I'm sure in this crowd we're all aware that teamwork is critical to safe and efficient care in the operating room, and there's been 2 decades of literature that have been showing the link between teamwork and communication and leadership to safety and efficiency in the OR. And along with that, we've also had a lot of research come out in the last 2 decades about team performance in the operating room and trying to understand those different domains, skill sets, and theories that contribute to a good team. So if it's so important in the operating room, how can we develop better teams? And so things like what you see on the slide, non-technical skills, psychological safety, leadership, conflict management, shared mental model, and many, many more that have all been really or decently well developed on their own. But what we lack is an integrated understanding of how all of these theories and constructs and skill sets interact in the real operating room environment to create that feeling of a good team where all things are clicking and things are moving in a coordinated way. And so we started to explore the construct of tone as a way of understanding how all of these team factors are interlinked in the real operating room environment. And so this has emerged more as a peripheral finding from many studies where they described this environment that's either tense or relaxed or focused in the operating room that kind of represents a shared understanding of team norms and expectations. It can be linked to safety events, and it creates this sense of an adaptive and coordinated team. But that's a peripheral finding, and it hasn't been formalized in the literature as of yet. And so the objective of this study is to formalize the construct of tone, including its definition, antecedents, and consequences. And to do that, this is a qualitative study. So we interview surgeons, nurses, anesthetists, and perfusionists, and we discuss their experiences working in a team in the operating room, having them reflect on their experiences when things went well and when things went poorly, and specifically with the lens of tone. So our participants define what the tone is and describe their experience with how that shapes teamwork. And this is analyzed using a constructivist grounded theory approach. And so this is our working definition of tone. It's an affective team construct that emerges from team interactions and is highly steeped in contextual factors. And so I'll break that down into each of its parts. An affective team construct means that it's kind of a feeling. So when we hear people describe tone, they use words like jovial, tense, focused, frustrated even. It emerges from team interactions in the way that we know what the tone is, and the way that we experience the tone comes from how we see others interact with us and amongst themselves. And it's highly steeped in contextual factors in that the tone will be shaped by what the nature of the case is, if it's high risk or low risk, if complications emerge, who those people are in the team and how they relate to each other, and so on. And so as we started to explore what the tone was, we also saw many links to different team processes and outcomes, and I'll go through 3 of those now. So the first was psychological safety. So for those who may not be aware, psychological safety is the feeling for team members to feel free to speak up, ask a question, share a concern, with their group without fear of negative repercussions. And we found that tone was linked to psychological safety in a very dynamic way. So not just across one case, but within a single case, the way that the tone was set encouraged or deterred team members from speaking up. And we have a great quote from this perfusionist who says, if there's a positive atmosphere, people will speak up more. If there's something going wrong, or if they've done something wrong, they'll actually own up to it, right? And a lot of the participants actually linked that speaking up behavior to patient safety and patient safety concerns, describing that when they didn't feel free to speak up, sometimes they waited so long that the moment passed and then the mistake already happened when they had the opportunity to actually avoid it. We also saw that situational awareness was linked to tone. So situational awareness describes a shared understanding among team members about where you are in the case, what's going to happen next, what's required at that point, what could happen, and what's required of the team at that time as well. And so the tone also helps to shift situational awareness through the OR. You can imagine team members who cannot see in the operative field, the way that they're actually picking up on information is seeing you go as a surgeon from chit-chatting to quiet, and then they realize that maybe something's happened. You've entered a critical phase, you've had a complication, they start to gather more information, and then from then on they start initiating their new work procedures to support you in that way. And so this nurse says, I think the tone and voice is really important when you hear it. Even a slight volume change in their voice also kind of tells us or indicates, okay, this is something serious, we just need to concentrate now. We also heard that the tone affects enjoyment and quality of work over time in the operating room. And at first when I found this, I thought, okay, it's nice if people are happy at work, but does that really affect our outcomes in our system? And I think this one participant described it really well. He says, a positive tone makes those hard days seem not as hard. It makes you want to come into work and do your best, and it definitely makes the quality of your work better. But I think mentally it helps when you're having those really long days, you had a stressful situation, you lose a patient, that kind of stuff. I think that it helps to feel valued. And so we can definitely understand in the context of burnout in our healthcare system, setting the tone in a way that helps people feel valued may actually be very important. And so we started to see that tone is a construct that we can use to understand how teams interact in the real environment in a way that's not kind of divorced from the actual team context. And so then we started to examine the team interactions. I said earlier, it's rooted in the way the team communicates. And so later on in a study, we'll be examining what those people are actually doing to set the tone, but here from a theoretical perspective, we want to understand how people in the team are relating to each other to develop that sense of a tone. And the best way that we could organize these thoughts is through taking an interprofessional lens. And so I'll go through each of the roles of the different professions in the OR. So surgeons across the board were viewed as setting the tone. Surgeons, non-surgeon staff, whether the surgeon was aware of it or not, they were seen as setting the tone, and their demeanor set the tone for the rest of the team throughout the entire case. This surgeon says, I think that the surgeon role is being almost like a skipper, of a ship. We need to make sure that this ship is going well. It's really important when things go bad. Let's say when there's major bleeding or something unexpected happens, that's when really the skipper needs to lead the group. If the skipper's in a panic, everyone is going to be in a panic. So the surgeon is really central to keeping that team together in response to critical events. Their position as tone setter comes from hierarchy. So this perfusionist says, if you look at the hierarchy in the room, you have surgeons, anesthesia, and then everybody else. So the anesthetist could certainly set the tone as well, but to a much lesser degree. And speaking of anesthetists, they described a different role. They didn't set the tone, but they modified the tone. And the best way I can explain it is to show you another quote. This anesthetist says, I change the tone for sure. Sometimes I come into a room and it's just paralytically serious. I will lighten that mood a lot. Sometimes I find it's not serious enough, and I'll make sure it's more serious, and I'll make sure that the surgeon understands that I'm concerned about this situation, right? So I don't try and set the tone, but I try to modify the tone as best I see it. Their ability to modify that tone, again, occurs in the context of hierarchy, and this is kind of a funny quote from a nurse who says, I could see anesthetists, if they sense a negative tone, they try to make a joke, and then if the surgeon still responds in a more serious tone, then they just shut up. And so their ability to really modify that is limited by, again, that surgeon who's ultimately in control. The relationship between the anesthetist and the surgeon has also come out as a very important variable in this tone-setting environment. So this anesthetist described a time that was very tense in the operating room where she spoke up to the surgeon, and when she ended the story, she said, but he's a good friend, so that's why I said it too. I wouldn't have said that to someone I don't know well, right? So maybe we can rely on our friends a little bit more to keep us in check, but when we have somebody in the room that we don't know, we should be a little bit more aware of the tone that we're setting. And lastly, we'll go on to the nurses. So they didn't see themselves as setting or modifying the tone, but they facilitated the tone. They described certain things that they did that promoted a certain tone from the surgeon. Their degree of preparation or lack of preparation kind of set the stage for a certain tone to be set, as well as the confidence that they displayed over time. They also support the tone that the surgeon sets. So this nurse says, it's part of our job to kind of go around the room and say, okay, this is quiet time, something's happening right now, let's all pay attention, the surgeon needs to concentrate. So they're supporting that tone. And so through our study, we've started to see that tone is an important team construct that can help us link all of those different factors to understand how team dynamics play out in the real operative environment. And so as I continue this study, I reflect on these two questions that I invite you to reflect on as well. First is, how can we leverage tone setting to create safer, more efficient operating room teams? And more specifically for the surgeon, how can surgeons promote a tone that's conducive to high quality teamwork? And so I'll leave it there, and I'm happy to answer any questions.
Very good presentation, and as you know, I'm very interested in what you're doing. Dr. Yasufuko is going to be the discussant.
Thank you very much, Hillary, for an excellent presentation. I will have to disclose that I think I was interviewed, so one of the comments might be my interview. I think teamwork is very important in the OR, and I think all the surgeons in the room know that. We try to set the tone in a good way, but we're not always good at that, especially when the S hits the fan. I think people react differently. And I think your study of tone really explains that, I think. And but we're kind of talking about that surgeons need to set the tone, but is it really the surgeon that needs to set the tone, or do you think the entire team needs to set the tone?
I think what we've seen through the study is that certainly the entire team is responsible. There are those different roles, and they all have a role to play in coming to the operating room prepared to be a team player. But what we have heard from non-surgeon staff is that whether the surgeon is conscious of it or not, their mood and the way that they're interacting will set the tone because of their role in the hierarchy as the most responsible person in that room. And so it is kind of an added cognitive load, or an add to your cognitive load, but it's something that I think we should all recognize as part of a surgeon's expertise and kind of take control of that role as leader in setting the tone.
Thank you very much. And I think it's really a teamwork. So I feel that it may not just be the surgeon setting the tone, and especially what I found very helpful is when we do the checklist at the beginning of the case, we really gather as a team, and I think that's where we can all set the tone for the case. So I like that teamwork, approach to things. But having said that, how do we teach maybe surgeons that do not set the tone appropriately? Is your study going to help us understand? How do we intervene those surgeons?
Yeah, I think one of my goals with this study, I have kind of several goals that maybe would span my entire scientific career, but my first goal is just to raise awareness of this topic. We heard in our interviews from more senior surgeons that they didn't even become aware of their role as leader in the team and the effect of their behavior until much later in their careers. And so I'm wondering if we can use this understanding and just at first creating a shared understanding of this team dynamic in the operating room to shift that awareness a little bit earlier. It's challenging, of course, because new surgeons are very stressed out about the technical aspects of their operation. But I think the first step is just to raise that awareness. And then later on, the next study that we're working on is to actually look at the specific behaviors within each person's personal style of leading the team, which behaviors are most conducive to teamwork and which ones aren't. And hopefully we can work towards helping surgeons maximize those more positive behaviors and minimize the negative ones.
Yeah, thank you. And lastly, it's just a comment, but I think your research is very important. I haven't seen research like this, and hopefully the findings will help train surgeons that may not be setting the tone, in a good way. So I look forward to further research from your group. Thank you.
Thank you, Dr. Yasufuku.
Can I ask you also one question?
Yeah.
Do you have any plans to, like you did your research and you saw that this is what's going on. Do you have any plan to do any simulation with the team, multidisciplinary team, like anesthesia, nurses, perfusionists, surgeons?
That's a great question. We actually have started now recording real data in the operating rooms where we're recording mainly audio data, and the plan is to look at computationally, my background is in computer science, to look at computationally the patterns of interaction that are associated with different tones. So drawing upon natural language processing and machine learning, can we actually understand these team dynamics in a computational way that's informed by the theory that I presented today?
Thank you. Dr. Swallow?
I think Dr. Hamstra's first.
Thanks, Carol. Thank you. Great research. I think you're onto something with this idea of tone, but as an educator, I'm always cautious when someone comes up with a new construct, and because is it robust? So could we replicate this in different situations and circumstances? So I'm wondering if you'd looked at other fields. Like I would think that in aviation, like in the cockpit, tone might be something important. And the second question would be, does tone actually affect the degree of learning or the quality of learning of residents?
So for your first question, there are a lot of team studies that happen outside of medicine for sure and outside of surgery, and so we've drawn upon those theories. So not necessarily, I don't think that there's been a tone in those fields, but conceptually tone as a dynamic and emergent state is something that we've drawn from those fields. And incorporating the emotional literature, because if we're relying on team interactions, presumably the affective component of that is important. And so trying to draw in those theories and that work to inform our study of tone. And then I'm sorry, what was your second question?
Does it affect the quality of resident learning?
Oh, yes. So that was something that we separated early in our study, and I think that we have heard that surgeons are trying to set the tone for their trainees to learn and so on. We decided to focus on what I nicknamed practice tone, which is the tone for the overall operating room team. But I hope that in future we can kind of switch gears and look into the learning side of it.
You're probably familiar with Julie Allay's work on the surgeon anesthetist dyad and the 90-day morbidity outcomes being different depending on how familiar the dyad is. So I just want to sort of acknowledge that in the past 3 years, we as surgeons have lost a lot of anesthetists. We've lost a lot of nurses that we're used to working with, and it's created even more of a stressful environment. So I sort of like the comments you made about the cognitive load we already have, and then you add that onto it and say we're responsible for the tone as well. Just to sort of say on behalf of all of us, it's a lot already, and I think tread carefully, but I can see you're already sort of thinking that way.
Some reluctant leaders sometimes.
Good.
Thank you, everyone.
Our next presenter will be Dr. Marcy, who is part of the SSTP program, a PGY4 resident in neurosurgery, pursuing PhD through the SSTP, as I said. His talk will be about closed-loop decoding and control of attention in children.
Thank you very much. So I'm Nabras Warsi. As you mentioned, I am a senior resident in neurosurgery working with Dr. Ibrahim at Sick Kids. And I find, because I have a W name, attention deficits are always something that I get to sort of attract to myself. So we'll see how this goes. So one thing that I learned as a neurosurgery resident and a member of the SSTP is to perhaps find a project that you're so passionate about that you don't mind all the gray hair. And so as I work towards looking like this towards my graduation, I'm excited to share what I'm up to with you. So what's really important to me is the devastating impact of attention deficits worldwide, and not really something that we think about necessarily as surgeons treating. And the reason for this, or for my interest, is that attention deficits account for nearly 2 million years of life lost to disability worldwide. And furthermore, they impair academic performance and erode future potential in thousands of children. And what we really think about attention deficits, it's not just ADHD. So we see this in children with epilepsy, we see this in children with oppositional defiant disorder, and even other conditions that you may not necessarily think of as associated with deficits of attention. The most important thing for us as surgeons is that these are generally highly treatment resistant. So up to 50% of patients do not respond to psychotherapy, to Ritalin, to other sort of medical interventions. And so that really brings us to a point here where no existing therapy targets the dysfunction and the cause of attention deficits, which is dysfunction in neural circuitry in the brain. And so the way that I wanted to approach this is to actually study the human brain in vivo, and something that I'm very passionate about and lucky to do through the SSTP as a neurosurgery resident is to have access to these intracranial recordings of the brain. So what you see in this lateral skull X-ray is SEEG, or stereotactic electroencephalography, where we can very precisely reach and target parts of the brain that otherwise would be inaccessible. And so now we have the ability, through children with epilepsy who undergo this procedure, to locate and map epileptogenic foci. We can also study how the brain works in such an intricate and deep level that was absolutely not possible before. So the first question that I was really interested in asking, and some of the data I'll show you today, was whether there is a core circuit in the brain, if you think of this organ as a large kind of circuit diagram, that's responsible for controlling attention and perhaps predisposes to deficits in attention. So how I did this, we recruited 28 children who were undergoing SEEG surgery for seizure mapping at Sick Kids. And what you'll see in this sort of little schematic is the children on postoperative day 1 kindly agreed, and often they didn't have much else to do, so we made it kind of fun for them, to do this task of flexible attention. So what they had to do is on this computer screen respond to different stimuli. And we looked at their intracranial brain activity, and we also looked at where they were looking. And what was critical here is we used computer vision and deep learning, and I devised a convolutional neural network. And what that did is it looked at the brain waves before every stimulus, those little shapes that the children have to respond to, was shown. And we tried to see if we could use those brain waves, to predict if the children were paying attention, so they had a fast reaction time on this task, or if they were going to lose attention and have a slow reaction time. And so what we achieved in our 28 subjects was an AUC significantly above chance. And what I'll show you is really cool about this is, number one, this is the first time really in human beings that we can use brain activity to predict attention and figure out if you're going to lose attention before it occurs. So I know if you're paying attention to me right now or not. Furthermore, the other thing that we did is we did the testing, the training of this model on postoperative day 1, but then we waited for another day or two between which there were changes to the anti-epileptic drugs, the children got more or less sleep, there were other interventions that were going on in the hospital, and we only tested based on the second day data. So it had never seen this model, these fluctuations in brain activity, and therefore they're quite robust. So next I wondered, now we can predict lapses in attention before they occur. But what is the brain actually doing in order to predispose to these problems? So what I'm showing you here are the main brain changes that we converged upon. And at the top, you'll see the letter theta for the theta band in EEG, which is slow frequency brain activity. And you see that essentially the frontal lobe slows down when you're about to experience a lapse in attention. And furthermore, you see that there's a decrease in high frequency beta band activity. So there's this shift in balance where now your brain is potentially sort of thinking or oscillating more slowly, and that's also why you lose your focus. Now, the reason I got really excited about this is we only have one known biomarker of ADHD, and that is indeed an alteration on scalp EEG of the theta and beta ratio. So what that suggests, because most of these subjects, yes, they had epilepsy, but they did not have ADHD, or most of them did not. This suggests that from moment to moment, even your brains, when you're losing focus, may actually resemble the brain of children or adults with ADHD. So next I talked about the brain in terms of circuitry. So I showed you these brain regions that were sort of so accurately predictive of attention. But what I wondered is what the brain circuit was that all these sort of regions connected to. And so we looked at this connectivity first in a large human connectomic project dataset of 1,000 patients. And what we see is really a network lit up. And that network is the network that predicts lapses in attention in these children, but also by looking in this large adult dataset, presumably in broader populations as well. Now, the network looked the same in the fMRI bold resting state connectivity of the children with epilepsy. And moreover, in a completely separate dataset of kids with ADHD, we saw that not only was this network predictive of lapses in attention, but the connectivity in the circuit actually predicted how severe the ADHD was going to be in these completely separate subset of patients. And so to segue to my last sort of main finding, of course, from a surgical perspective, we wondered, now that we found a network, can we intervene on this network and perhaps be able to control or improve attention? And so what we did is the children performed the task one more time. This was generally on postoperative day 3 or 4. And now this time I took those machine learning models that I had trained to accurately predict lapses in attention, and I deployed them in real time. So what happened on every single trial was the machine learning model output a prediction, whether the child was going to be paying attention and have a fast reaction time, or whether they were going to be slow and basically be inattentive. And what happened then is this allowed us to complete a randomized intent to stimulate analysis. So we had 12 subjects where an intent to stimulate is recorded when you're about to have a lapse in attention, and no intent is recorded if we predict that the children were paying attention. And what happened then is that in 50% of the intent to stimulate cases, we delivered stimulation of the anterior cingulate cortex, which is a key part of this attentional control circuit that I told you about. And the other 50%, we withheld that to have an appropriate control. And finally, there were some trials where the children were paying attention, where we did not stimulate. One additional control is we looked at what happened if we stimulated when the children were predicted to be paying attention, and we termed this mismatch stimulation. So what happened here, I mentioned eye tracking earlier on, because eye tracking really is the sort of the window to the brain. And what we saw is during a lapse in attention, when we stimulated, the children looked at the target regions of those stimuli they had to look at. And so the green shows statistically significant results where they're now actually looking and focusing directly where they need to be. And orange is where they would have been looking were they not stimulated. And we also looked at reaction time, our main measure. So you'll see basically a baseline reaction time for every subject. And when the models predicted on this third session that the children were going to be fast, they were indeed fast. When the models predicted they'd be slow, they were slow. And now in a lapse of attention, when we stimulated them, all 12 subjects significantly improved and actually performed the task and indeed paid attention better. We looked at the quartiles of the reaction time here to make sure this was not some outlier effect, and certainly was not, which of course is good for me. And what this really showed us is that not only through reaction time, but also through the movement of the eyes and really the window of the brain to attention, we can greatly restore sort of attentive ability with brain stimulation. I also mentioned that we looked at the effect of stimulation during periods of predicted attention. And so what you see here now is when you don't stimulate in good trials, the children are looking at the target stimuli as they should. But essentially, if you stimulate at the wrong time, in orange, they look away. And furthermore, they're significantly slower on the task when you stimulate and they're paying attention. So this essentially proves that not only can we use stimulation to enhance attention, but we can also use it to make it worse, which is the sort of key construct of attentional control. And the last thing I wanted to show was to just touch on generalizability. So of course, as a neurosurgery resident with an interest in epilepsy, I'm happy to talk about putting electrodes in and doing all these types of things. But we wondered if these findings from subjects with epilepsy generalized to broader populations. So I'll show you here a scalp EEG example, not from intracranial EEG, but of what essentially the scalp brain activity looked like in our children when they experience a lapse in attention. And these red regions essentially show that the middle and the front of the brain are slowing down. Now, when we did a similar task in nearly 40 healthy adults, we saw the exact same signatures where, again, there's this frontal midline increase in theta band activity. So we wondered now, could we use non-invasive stimulation of presumably the same network, but through a surface node and achieve the same effect? So what we did is we did TMS, transcranial magnetic stimulation, of the region where we saw the greatest difference, which was also a node of this attentional control network, but of course without a surgically implanted electrode. And we saw that when the theta power was high, or when these people were experiencing a lapse, and we stimulated, they significantly improved as well. And so essentially this proves that the neural signatures of the brain slowing down during an attentional lapse that we identify are generalizable. And beyond that, we can potentially use both invasive means through surgical operations and also non-invasive stimulation to control attention in human beings. So to sort of summarize, there are two major first-in-human findings of this work. The first being that we identified a human attentional control network that was responsible for predicting lapses in attention and also can predict the severity of clinical ADHD. And furthermore, we show that modulation of this circuit, whether through invasive or non-invasive stimulation, can directly control attentive behavior in humans. So the takeaway points here are that we hope to redefine the treatment of children with attentional disorders, both through invasive and non-invasive therapeutics. And certainly there are several ethical, moral, and legal implications whenever we get into the realm of psychiatric surgery and the potential for human enhancement. And that's it. I have many acknowledgments to make, most importantly, Dr. Ibrahim, my supervisor, for allowing me to do this work.
Thank you so much for the fantastic presentation. Dr. Drake is here to discuss it.
Nebras, fabulous
Hi, Dr.
talk,
Drake.
and a great example of how you can get an amazing amount of information and do a very important study when you're doing a test for something else, such as SEEG. And related to that, SEEG is very precise, but it also samples a very small part of the brain. And some of the areas you're talking about here, like the frontal lobe and the anterior cingulate cortex, they're large structures, and there aren't really specific targets I'm aware of. So how do you think that might have affected the results that you're getting here?
Absolutely. So actually something I was hoping to talk about, part of the reason for deep learning, so this ability to analyze so much data at once, was that there is so much heterogeneity in where the electrodes are placed because they're placed for the purposes of epilepsy monitoring. And every child may have a different seizure focus. So the reason we used this approach was to converge on a subset, a very small number of brain regions, so only 8, compared to the thousands of different sites that these electrodes are put in. So one thing was sort of bringing it down to specific regions with the machine learning. But the other thing is I also looked at sub-regions in particular that were important. And for the anterior cingulate, it's especially stimulation of the dorsal component, which is known to have a more sort of neurocognitive role than, for example, the more rostral part that is commonly targeted for things like depression, anxiety, and what have you.
Thank you. So for your machine learning algorithm, you showed a receiver operator curve, and it was significant, but it was 63%, which is not like the most robust. So how do you think using that as a discriminator for your subsequent tests might have affected your results?
Yeah, so this is part of the reason came into this question of whether there is harm to be done by stimulating at the wrong time. And so that is why, with this AUC of about 0.63, 0.65, we wondered, well, if we stimulated at the wrong time, did it do nothing, or did it potentially make them worse? And so what I think is nice to have an additional sort of control there is that we found that when we followed what the model said, we certainly made them better. And when we reversed what the model was saying, we made them worse. So kind of the short of the long is that I think it works well enough at this stage, but there are certainly ways to make this better. One of them is if we were to do this as an intervention or a clinical trial, we'd pick regions to specifically target rather than having to sort of have a bit of the heterogeneity in the epilepsy implantations. So that may help.
And my final question has to do with the ethics of this. And not everybody thinks ADHD is such a bad thing, right? And it's been associated with increased creativity. And some of our most famous scientists and entrepreneurs are said to have had ADHD. So just talk to us a little bit about that and how this therapy might be implemented.
Yes, so I think the big thing whenever you think of psychiatric surgery per se, it has kind of a not so great history when you go back. And I think that's another caution in terms of why the ethics is so important. And when I look at this, I really think of this as if you're going to proceed to a surgical intervention, it's for debilitating, severe, very last line cases. So really a situation where medications don't work, psychotherapy doesn't work, and certainly the patient is interested in pursuing the intervention. But with research studies, you also have to be careful in terms of what you offer when you're testing things out. So I'd say caution is certainly warranted. And certainly I look at this from a surgical standpoint, but when I talk to my friends about this, some of them say, can you just make an attention helmet with a little TMS coil in it, and we just put it on and focus better. So I don't know if that's what I'm into, but that's certainly another approach.
Okay, thanks very much.
Sorry, I almost had the same point. I'm reminded of Dr. Yang's talk earlier today where he showed a slide of him staring into the sky. Right?
Which is an important thing to do sometimes.
Thank you very much.
Thank you very much. Thanks.
Our next speaker is Anya Gopita-Nair. She's a third-year medical student at the University of Toronto. She completed her undergrad in McMaster University. She will talk about the contemporary trends in breast reconstruction use and impact on survival among women with inflammatory breast cancer.
Hi everyone, my name is Ananya and I'm a third-year medical student at the University of Toronto. Thank you so much for the opportunity to present our research here today titled Contemporary Trends in Immediate Breast Reconstruction Use and Its Impact on Survival Amongst Women with Inflammatory Breast Cancer. This work was completed with the support of Dr. David Lim, Dr. John Semple, Dr. Stephen Nerode, and Vasily Giannakis at Women's College Hospital. So I have no financial disclosures or conflicts of interest with this presented material in the presentation. So just for background, inflammatory breast cancer is a specific type of breast cancer that's quite rare. It's about 1 to 5% of all breast cancers that's diagnosed. It's typically associated with younger women, less than the age of 40 years. And it's a clinical diagnosis made by the peau d'orange appearance, which is characterized by swelling, redness, tenderness of the breast, along with pitting or thickening of the skin, retracted or inverted nipple, and general pruritus as well. It is a hard to diagnose breast cancer given it is a clinical one and it's often missed on mammogram as well. And it does have a significantly worse prognosis than other breast cancers due to its aggressive nature and early lymphatic involvement, typically developing over a course of 3 to 6 months. So given the aggressive nature, typically by a lot of the time it's diagnosed, women have 1/3 of women are actually metastatic when they present for inflammatory breast cancer. Given all of this, immediate breast reconstruction is traditionally discouraged in women with inflammatory breast cancer due to concerns about recurrence, poor long-term survival, and possible delays to adjuvant radiation therapy. And in the past, over 5 years, survival rates were less than 10%, with recurrence rates of 65%. However, with advances in trimodal therapy, which is the combination of neoadjuvant chemotherapy, followed by mastectomy and then radiation therapy, there have been significant improvements in oncological outcomes. For example, it's now closer to 30 to 70% in certain studies. There is new literature also suggesting limited association between immediate breast reconstruction and worse oncological outcomes in this population. So our objective specifically was to determine the contemporary trends and predictors of inflammatory breast cancer reconstruction in these women and whether or not there is an impact on oncologic outcomes. So to do this study, we did a retrospective study using SEER database from January 1st, 2004 to December 31st, 2015, so over 11 years. The inclusion criteria was adult women who had non-metastatic inflammatory breast cancer. We excluded women if they were above the age of 85, if they had a previous cancer diagnosis or a previous cancer history, if they were of unknown stage, or if they received some form of breast conserving surgery or no mastectomy. We collected demographic data, breast cancer specific data, along with surgical treatments, and we defined follow-up as following over the course of the diagnosis until they were discharged from follow-up, along with any death that has occurred due to breast cancer or other causes, or loss of follow-up. One thing to note was that for our SEER data, unfortunately, we could not get delayed breast reconstruction information. It was limited to immediate breast reconstruction. So when we were analyzing this data, we did a twofold analysis. The first one being we did the crude breast cancer analysis comparing reconstruction with no reconstruction. The second was we did a propensity score matched analysis where we compared 1 woman with reconstruction to 3 women without reconstruction. Women were matched in the scoring system based on year of diagnosis, age of diagnoses, the surgical procedure that they underwent, and the propensity score itself was demographic data, tumor characteristics, and surgical data as well. So overall in our results, we had 4,076 patients who underwent a mastectomy included in our study. Roughly 10% had immediate breast reconstruction, and 22% also had a bilateral mastectomy as well, mostly in the name of a contralateral prophylactic mastectomy. The women in our population were aged 55 years. Predominantly, they were Caucasian. They lived in metropolitan areas and also had a higher median income and access to private insurance. The mean tumor size was 6.1 centimeters. There was significant nodal involvement from vast majority of women, and they were all vast majority also had high-grade tumors. Some of the differences we initially found between these two categories of women, the ones who had reconstruction and did not have reconstruction, were that the immediate reconstruction group were younger, they lived in metropolitan areas, and also had a higher median income. However, interestingly, there was no difference in the tumor size, in the nodal status, or the ER/PR HER2 status of these women. So unsurprisingly, over the course of the 11 years, immediate breast reconstruction did increase from 6.2 to 15.3%. And the factors I mentioned earlier were predictors of immediate breast reconstruction. And again, surprisingly, race, tumor size, grade were not predictors of women undergoing breast reconstruction. Similarly, contralateral prophylactic mastectomy rates also increased during this time from 12.9% to 29.6%. So when we did our analysis, the crude 10-year breast cancer-specific survival found that women who actually had breast cancer had improved survival at 62.9% than those who didn't have reconstruction, which was at 47.6%. But this is likely given that these women were younger, they had differences in their socioeconomic status, maybe year of diagnoses. And to kind of account for these factors is why we completed the adjusted propensity score analysis. And at this point, we found that there were no differences whether or not the women underwent reconstruction or they chose to not have reconstruction. So it's interesting that, unsurprisingly, that more and more women are opting for immediate breast reconstruction, and it coincides with improved therapeutic options. Survival over the past decade have also improved, as seen with our 10-year rate of 56.6%, which is quite different than the previous rates of less than 10%. And given the improved longevity of many of the women, there might be consideration to be given to the impact of immediate breast reconstruction on the psychosocial well-being of women and the quality of life that one may get from breast reconstruction. There are some limitations, however, specifically the retrospective nature, and there's also some gaps in the SEER database that we had access to. Specifically, as mentioned earlier, this is all on immediate breast reconstruction and not delayed. We didn't have access to complication rates, which is typically more associated with immediate breast reconstruction. The type of reconstruction itself, whether it was alloplastic or autologous, we couldn't differentiate in that case. And there are some details about completion of chemotherapy and radiation therapy, which was missing. But overall, immediate breast reconstruction after mastectomy can be an acceptable option to women if they so choose, as it's not associated with worse oncologic outcomes. However, further research is needed in determining who the optimal candidates might be who can appreciate the larger benefits of undergoing an immediate breast reconstruction. Thank you so much for your attention, and if there's any questions, happy to take them.
Thank you very much, Excellent. Dr. Melinda Musgrave, please.
Hi, thank you so much. I love this paper. I'm a big fan of immediate breast reconstruction of locally advanced breast cancer, but even I've shied away from inflammatory. And the current Ontario guidelines say we should wait 2 to 3 years. So I'm hopeful with this paper that we'll see these women in a better light. I think your data mirrors most of breast reconstruction, younger women, more socioeconomic status, bigger metropolitan centers. And I think the reasons for that in Canada are the same as in the US. The financial stuff is different in the US. There were a couple of interesting things I saw in your paper. You showed that bilateral mastectomy, so contralateral prophylactic, doubled at the same time during the immediate breast reconstruction phase. Why do you think that happened?
I guess with younger women, particularly if they are also undergoing reconstruction as an option, it could be aesthetics related. And typically if they're also diagnosed younger, the odds of, the fear of having a it showing up in another breast could also be something that's considered as factors and could be pushing patients to undergo a lot of prophylactic mastectomy, even though I believe the data doesn't necessarily suggest that it is beneficial, but I guess it's the psychosocial aspect that kind of comes into play at that point.
I think the most important thing you did was the adjusted 10-year survival. Tell me about that and tell me what you would wish to happen for this paper with respect to women in those groups, the LABC type groups, getting breast reconstruction. Yep, so your adjusted 10-year survival really shows that whether I get my reconstruction or not get my reconstruction, I'm going to get my cancer back or die from my cancer at the same time. So why shouldn't I get reconstruction as someone who has inflammatory breast cancer? So what would you wish for people to get out of your paper when they read that, what you want to happen for those women?
I guess it kind of comes back to, it's something that also that can be presented to women during that conversation of what treatment looks like. And that's kind of where the next steps of this research in terms of psychosocial well-being will come into play, because there is significant benefit when you undergo breast reconstruction for certain women. And if certain women choose to have that either from a cultural standpoint or marriage, whatever else it is, that can be a conversation that's more openly had. And maybe clinicians may feel more confident in approaching that conversation if they know that there's research that's building or supporting whatever conversations and decisions they're having.
And one final piece, you said race did not come out in your data, but 79% of your women who had immediate reconstruction in the IBR group were Caucasian. How do you explain that data?
That's definitely something that we wanted to also, that we recognize in the data, that it's not capturing the larger variety of women who have inflammatory breast cancer, because there are definitely cultural differences that are associated with even race as well, who may choose one way or the other and how they view their own health. So that's something that we definitely need to investigate a little bit more, and maybe do a sub-analysis of just the women who were people of color to see if there's a difference in how their decision-making plays out.
Thank you for this paper.
Thank you.
Thank you very much. Excellent.
Thank you so much.
Thank you, Haya, for chairing this afternoon, and thank you for the speakers, fantastic, and for all of you die-hard surgical researchers here, and for sticking around. Well, what a great day. And we're just wrapping things up, so I'll make a few comments, and I'll hand over to our Chair, Carol Swallow, for some final comments also. So you know, we had a fantastic symposium today where we were kind of exploring really kind of the technical frontiers of where surgery is going. And I think we did it in a very interdisciplinary way that I think highlights the strengths of the department, you know, thinking about artificial intelligence, advanced imaging, robotics, from really very basic levels right out through a translation. Like, fantastic. Professor Alan Lumsden, amazing talk. I mean, people were riveted. It was just absolutely amazing. And although Roz Bradford was not able to be here, I know that she would approve, and she would be telling me this at dinner, that I did an okay job of selecting the Gordon Murray lecture, and that her dad would be proud. So thank you very much for that. So big thanks to Renata Musa for organizing a special thanks to the whole research committee, all of the chairs, all of the faculty that were judging and you know, contributing their efforts. And in particular, shout out to Andre McCart for a lot of effort in support of today as well. Big thanks to all of the trainees, fantastic. So the surgeon scientist trainees and the trainees who are not in the surgeon scientist training program, but who also contribute in an amazing fashion to all of the surgical laboratories. The research was absolutely spectacular. I've probably forgotten somebody, but not sure. So in any event, I'm going to hand over to Carol. We hope to see you all at dinner tonight. Big thanks to Carol for all of her support, and closing remarks from our chair.
think I have 90 minutes, yes? So I'll take 90 seconds. Like he said, I think we saw today that Gallie Day is back, which is fantastic. It was just so exciting for me to have a full room. And I think the program was really well put together and well thought out. So thank you to Michael, thank you to the members of the research committee, and thank you to all of you for staying here and listening to our young trainees give it all they've got. It was so impressive to me. I'm really proud of this department. Thank you very much.